Introduction.
Hi, we are from the Institute of Biomedical Science at the Federal University of Rio de Janeiro in Brazil.
In this video, we'll be preparing chromosome spreads of human embryonic stem cells to detect the possible numerical chromosome alterations.
Although human embryonic stem cells have been shown to present a stable diploid karyotype, many studies have reported that depending on culture conditions, they become prone to acquire chromosome anomalies such as addition of whole or parts of chromosomes. The characterization of the genetic integrity of HASC in vitro is crucial, considering HASC as an essential tool in embryogenesis studies and drug testing. Furthermore, for future therapeutic purposes, chromosomal changes are a real concern as it is frequently associated to carcinogenesis. So, let's get started. Treating the cells with cosamide. In this procedure, we used the 89 HASC line, cultured in the MNF12, supplemented with the 20% of no-count serum replacement and 80 nanograms per milliliter of fibroblast growth factor. Begin by removing the culture flask from the incubator and under a laminar flow hood, add cosamide at final concentration of 0.1 microgram per milliliter. The addition of cosamide to the medium induces mitotic arrest in metaphase, when the chromosomes are well condensed and can be more easily visualized under microscope. Be sure to use sterile techniques to avoid contamination. When you have added cosamide, cap the flask and return the cells to the incubator. Fixing the cells. After the incubation period, the cells are now ready to be fixed. At this point, it's not necessary to work under sterile conditions.